All right, well, it's a pleasure to be here today. Thank you for the kind introduction. It's a very interesting symposium, I think, in terms of trying to think about this intersection between cancer and neurodegeneration. My lab does a lot of work on both, so we do a lot of work in cancer, we do a lot of work on neurodegeneration. We don't do a lot of work at the interface between the two, so this was sort of an impetus to think about that a little bit more than we usually do. So I'll start with the obligatory disclosures. I am a co-founder of a company called Aerovale, and I'm going to talk just a little bit about the project that led to that today. So in interest of disclosure, they did help fund that project, and then I'm involved with a few different pharmas and centers and such. That doesn't relate too much to today. So in cancer and neurodegeneration, of course, is the whole point of today's meeting. There's a lot of overlapping interests. I thought, we've got great insights into this from the early speakers. I really loved the diagram that Jane showed that really had, all the commonalities and then the things that are opposing as we think about cancer and neurodegeneration. I'm going to go through a number of those as we go through the talk today. There's an interesting metabolic basis to this. There seems to be a lot that is related in terms of overlapping genes. I'll go through a number of those, and we'll go from there. I also wanted to just say a couple words about just the brain in general, because I think we're focusing on neurodegeneration, and then of course there's brain cancer and all the other kinds of cancers. The brain really is a fantastically interesting frontier, and especially being here inside the Allen Institutes, where you've got the Brain Institute, which has been so influential, the Cell Biology Institute that obviously is putting forth all these cell types in the brain as well. Coupled with the artificial intelligence that's trying to recreate the ability to make a brain, makes this, I think, really just one of the singular institutes in the world. And I think that this whole focus around neurodegeneration and cancer and that interface is really helped by all of those other kinds of emerging elements here as well. So I'm going to talk a little bit about one of my major projects, kind of my major effort here just for a little bit, to give you a sense for where I'm coming from and what we're really doing. And almost everything I'm going to talk about today will be work from our lab in one way or another. And I just want to give a bit of a background and then bring it back to implications for cancer and neurodegenerative diseases. So I work in a field where we're trying to think a lot about what really drives health for individuals. And if you take this is taken from a 2007 New England Journal of Medicine paper, but if you look at the determinants of health in the United States and what really drives health over the span of your life, it turns out that about 30% of your lifetime health is attributable back in one way or another to your genetics, the hand you've been dealt. About 60% is attributable back to behavior and lifestyle, and we heard a lot about that in the earlier talks, especially I would say in Jane's talk about some of the metabolic lifestyle factors that implicate both the likelihood of neurodegeneration as well as the likelihood of cancer. And it turns out that only about 10% of a person's lifetime health is attributable back directly to the healthcare system. So that's a really strong reason to focus more on that 90% than what we do on the 10% today. So if you think about that 10%, we currently spend $4 trillion a year nearly in the United States on healthcare. And by the way, from a National Academy of Medicine report from a few years ago estimated that 97% of those costs are spent on care after illness. So one of the other things that I'll argue for a little bit here as well, and I think is alignment with some of the earlier talks, is a need to focus on those earliest stages of what's driving the generation of cancers and the generation of neurodegenerative disease. Conversely, we have a wellness industry that is focused on prevention. I'm wearing my Fitbit like a lot of people here and so forth. And there has been, but the wellness industry today, I would say, is a pretty mixed bag. There's a ton of it that's really fantastic, and there's a lot of it that doesn't have much of a scientific basis. And so this has led us to really push for what we call scientific wellness. And I'm going to talk about that just for a moment. And basically the notion of scientific wellness is to try to be very proactive in the course of disease, trying to optimize health, minimize the chance of transitioning to disease, reversing it as its early stages, rather than waiting for catastrophic failure like we get in the late stages of Alzheimer's or cancer. The desire to get into this area led Lee Hood and I to announce a project in 2014 which we called the 100K Wellness Project. So this is a aspirational project where we said we're going to recruit 100,000 individuals, do whole genome sequencing on everybody, and then multiple times per year measure proteomes, metabolomes, clinical chemistries, microbiomes, wearable devices, and so forth to generate what we call dense dynamic personal data clouds. The idea of this being that we can generate a huge amount of information that's dense enough in terms of how much we have, dynamic in the sense that we can get it over a long period of time and at good regular intervals so that we can look for the early warning signs for diseases like cancer and neurodegenerative disease. One of the things that I'll argue is that we often don't study what we most want to know in science. I'll use myself as the example. So I have a grant from the NIH. It's on Alzheimer's disease. It's with the Accelerating Medicine program for Alzheimer's, AMP-AD. But basically, we study the brains of people who have Alzheimer's and those who don't. But when do we do that? After they're dead, right? Nobody's going to give you a piece of their brain before then. And so basically, that's not what we really want to know. What we really want to understand is what was happening in the body of the person that got Alzheimer's disease or the person that got cancer 20 years before that wasn't happening in the body of the person that didn't get Alzheimer's. And what we really, really want to know is if we had known what that difference was, could we have altered it? Could we have changed that trajectory? So that's the notion here. And this project is basically taking two different forms. The first is, as I mentioned, this company, Aerovale, that Lee Hood and I co-founded along with Clayton Lewis, who's our CEO. And basically, this is a consumer-facing company. The nice thing about it is that if people come in through Aerovale, if they check a box saying that their data can be used anonymously for research, all that data now comes back into ISB. So this has been the way that we've financed this project. If you did a little math in your head for the 100K project, it's about a $10 billion proposition. So this is the way that we're making that happen. We've got about 5,000 individuals who are signed up so far. We've raised just under $39 million to get, or I'm sorry, just under $40 million to get the company off the ground. We've got 170 people working there now. And in essence, people come in a different financing model since they pay for the assays. But basically, the genesis of that is that you focus on scientific wellness, which is this immediate give back of information that has an impact for people's health in the moment so that they then contribute that data back so you can learn over the long haul and give them back insights into the early stages of cancer, the early stages of neurodegenerative disease. So it fills in a lot of that information that we don't get very often because we don't get super detailed measurements until someone signs up for a clinical trial or until they are sick. So that's how that's going forward. On the other side, ISB, if you're not familiar with us, ISB is a nonprofit research institute founded by Lee Hood in 2000. It's been standalone for most of its life, but this year, or last year I should say now, we're in 2017, but in 2016, we affiliated with Providence, which is the third largest nonprofit healthcare system in the country. So it's a 50 hospital system in the western United States. Lee Hood's become the chief science officer for the whole of the Providence system. We have a new incoming president now to ISB, which is Jim Heath, coming up from Caltech. But basically what this interface with Providence does is it lets us action on things that we can identify from these dense dynamic data clouds, launch clinical trials, do research of various kinds. And I'm going to tell you about a few of those studies in a moment that are actually both focused on Alzheimer's and cancer. So in the Pioneer 100, we looked at whole genome sequencing, so that gives you millions of differences in every individual. And by the way, this Pioneer 100 project, I'm trying to do a very abbreviated version of this, basically was the pilot for the 100K Wellness Project. And so we just recruited people, mostly here from Seattle area, the community, a few from other places, some of whom you would know. Won't get into that at the moment. So anyway, we did whole genome sequencing on everybody. We did the labs that I mentioned. So this was 150 clinical labs, 700 metabolites, 400 proteins, gut microbiome. These are all done 3 times during the year, as I mentioned before, and continual tracking. This gives us what we call our dense dynamic personal data clouds, and that's a database of actionable possibilities that grows over time. Okay, and I'll give you just a couple ideas from there. So one thing we can do is we can find new correlations across these data. And this is just a diagram that shows correlations that are statistically significant after multiple hypothesis correction that cross different labs. And the one area down here, and I'm going to go into this in detail, you'll see the, do anything? So if you look at this genetic traits at the bottom, there's only 108 individuals, so we're not looking at variants or anything like that with that sample size, but we can map to genetic traits. And I'm going to bring that up because what those traits are in general are disease risks. So you can take your disease risk for Alzheimer's disease or your disease risk for cancer, and then you can see what are all the other elements that are correlated with that disease risk and start to have an understanding of how disease risk manifests in the body. Okay, and I'm going to give you one example, actually not taken in this case from cancer or Alzheimer's. It's the one that's coming out of our paper. We have a paper on this that's coming out in Nature Biotech here pretty soon. But I will talk about one disease just to give you the concept, and then I want to relate that back to cancer and neurodegeneration, which will be the rest of the talk. So in one case here, we have a relationship. So these are just modules that we pull out in an unbiased way from the big network. I have some videos of how that happens, but I took them out. Especially after the first speaker, I don't want to be out-videoed by my crappy video compared to the amazing videos from Laszlo Barabasi. Anyway, but we have the cystine, and you'll see it's connected there to inflammatory bowel disease, if you see that right there. And so the interesting thing there is that inflammatory bowel disease in that case, it doesn't mean the disease. What's that a label for is for the genetic risk for disease. So one really interesting thing that we learned when we first proposed doing the 100,000 person wellness project, or we thought of that number of 100,000, and in talking about that, I should say Gustavo Glusman here in the room did a huge amount of work for this as well. But basically, when we thought about that 100,000, we were thinking about people kind of cascading down, and then you see a disease trajectory, right? And getting back to what's the trajectory into Alzheimer's, what's the trajectory into cancer, and so forth. And you need sort of a big number so that the number at the bottom, you have enough to learn something from. But in switching the paradigm here a little bit, what we found is that if you think about genetic risk, if you've got good GWAS mapping and GWAS variants, now every single person is relevant to the study of every single disease. Not everyone has a disease. Everyone has a genetic risk, whether it be high, low, medium, whatever it is. And so there's a lot you can learn from that. So one example that we did very early, and I just want to show the concept here. So we found that there was a strong negative correlation between, this is working at all? Yeah, a little bit, between inflammatory bowel disease genetic risk and the amount of cystine that's in a person's plasma. So the interesting element of that, and you'll see that's very significant even after multiple hypothesis correction, so 10 to the minus 6 even after you correct. But then we go into the literature, and it turns out there's a couple interesting things. One is that plasma cystine has been found to be one of the top candidate biomarkers out of the blood for IBD as the disease itself. And the second element is that ulcerative colitis, which is a form of IBD, turns out the lower your cystine is, cystine is a dimer form of cystine, basically the worse your disease is. And so what was interesting that there's just two facts, two implications there that I think are quite interesting. So one is that you can actually disambiguate what's related to the genetic risk for the disease from what's related to the disease having started or the onset of the disease. Because remember, we find this relationship with cystine in people who range in age from 20s to 80s and the range in age from 20s to 80s in which nobody has the disease. So that's one. That means you can do a correction factor for basically any case control study in the world, because this gives you the background that's related to the risk of the disease rather than the onset. The second major point is that you can think about pathways to prevention. So in this case, there's actually a pretty interesting hypothesis that comes up. So you have these genes, that genetic risk manifests as this lower cystine. Turns out there are other papers that show that suppressed cystine, which sits upstream of glutathione, means that you're less well able to deal with oxidative stress. Oxidative stress is a trigger for IBD, so it suggests something immediately, which is that you're at high genetic risk for IBD. If it's mediated through this, though, you could imagine a very simple sort of nutritional type, metabolic intervention that would change you from being at high genetic risk to being at low risk for that disease. So you can swap yourself. So I just wanted to bring up that concept briefly because that's a concept that we'd love to get into in a lot more detail around all the genetics that are known about Alzheimer's and all that are known as cancer. In fact, we have ongoing studies to do that, but we haven't quite taken them as far as with IBD quite yet. But basically, what you can do then is to start mapping for all those genetic maps that you saw earlier, right? If here's your very high risk but low penetrance genes to your high penetrance low risk genes. For all of those, you can start doing a mapping from those genetic risks onto how does that manifest into the body. What are the kind of things that are altered in the blood? And then by mapping that into networks, you can figure out pathways to, is there a way to intervene to make a difference in how that genetic risk seems to be manifesting? Okay, so I just wanted to put forth that concept because I think that's going to be really an important factor. I also want to say, obviously, we're trying to track individuals at early disease, and I'm going to give a couple very small n of 1 examples that just come out of this first year of monitoring. So in one person, this is a pioneer, this is someone who's coming through Aravale. He was diagnosed with, was not diagnosed. He was coming in, and basically we had blood draws for him. And what we noticed was, in essence, he had really high, if you look at his leukocyte level, so that's just a histogram of the leukocyte levels that are measured from every blood draw from every person that's come through our program up to this point in time. And what you can see here is that this person, here July 2015, we saw they were a very big outlier. November, we saw it again, so you can see this repeated big outlier, so we referred them back to their physician to go get that checked out. And in fact, what happened, you can see how elevated this is, was that they were in fact diagnosed with leukemia. So this was a person that had leukemia, didn't know about it, and so we were able to go back and anyway, just trigger that so that they knew about it, get it treated before it became a more major problem. Another interesting thing is if you go back into the proteomics data, so here's a particular protein, and again, this is everyone in the data set, and here's the pioneer with leukemia. So you can just see major outlier, so potential. This is a protein that's involved in the stimulation of B and T cells, and so basically this is something that would be, we now have marked to follow to see if this translates to something that might be a good marker and things like that. Okay, and I'll talk a little bit about that, debating with myself whether or not to regress. And we actually think that the way that biomarkers are done should be radically altered. I'm trying to decide if I want to get into that too much. I'll say a few words about that in a moment. One of the things is, and then here's another person. So this is a woman in her 50s, in this case pancreatic cancer. She was diagnosed with pancreatic cancer in January. Bert Vogelstein and others have argued that when you're diagnosed, on average, it means you've had that disease for 5 years. We went back in her blood. She'd been in our program, in this case, for 18 months. So we could go back to that old blood draw. Here's another protein. You can see here's the distribution for everybody. And here is her. So she's, again, major outlier in this protein. This is a protein in the Notch signaling pathway. It is expressed in the pancreas. It's related to pancreatic cancer and so forth. And so there's some interesting elements here. So one of the things that we're really interested in then obviously is monitoring people and trying to understand early warning signs for disease. And we talked about pathways, so pathways both to Alzheimer's, pathways to cancers and so forth. And basically, in terms of how biomarkers are done, it's one thing to see this sort of in retrospect, right? It's a very different thing to say, okay, I see this signal, so the next person I see high on this protein, are they in fact getting pancreatic cancer or not, right? So that's one way, and we're going to be following that to see how true that is. And as this really ramps up big, we have already automated systems that are being built to monitor all of this in real time. So the other thing, though, that's really fascinating is to not think so much about biomarkers for a disease, but actually to think about divergence from wellness. And what I mean by that is what we're working on right now is essentially having wellness states that are very well defined And then as you observe these outliers, essentially, or divergence away from wellness, you can then do an analysis of all of the things that are divergent and then try to do a mapping of that back to a disease or a different process or something like that. So that's different than saying that this protein is always what's going to be the signal. The idea, though, is that if you're developing a disease, you're going to perturb something away from normality. You should do an analysis on all of those things that are divergent. And if you've got these dense dynamic data clouds, you can then analyze all those things that are divergent. Where do they seem to originate from? Where do they seem to come from? It moves you away from having to think about something as being the biomarker for X into basically an analysis on the set of things that are divergent. So this is really where we're going on that. And I want to move forward in the interest of time here. So we are doing a number of projects related to what I just showed. We're launching a clinical trial. Jared Roach, who's here in the audience as well, is really driving this, along with a number of our collaborators, Dale Bredesen at UCLA being one of them, who has very much in line with what Jane talked about earlier on a metabolic basis for Alzheimer's, has developed a very holistic type of program that's based on shifting metabolism in different ways, exercise, brain training, hormones, very comprehensive approach. But he now has 120 case studies where he claims the ability to have reversed early stage Alzheimer's disease. So we're going to test that now in a prospective clinical trial. Providence just committed $10 million to this trial to really get us going on it. So we're excited about that. We could talk a lot more about that. We're also driving a scientific wellness program for cancer survivors. It turns out this is being done with Swedish Cancer Institute here in town, led by Tom Brown. But in this case, we're looking at people who have gone through, have actually survived cancer, but have a lot of negative consequences due to chemotherapy and things of that nature. A lot of that actually comes back to their brain as well. They talk about brain fog or memory loss and all these kind of elements. And so we're going to do a deep dive on that. And then another that we're launching of these, what we call translational pillar projects around scientific wellness, is that Providence has actually now put 2,000 of their own employees through the Aerovale program. That's going to give us all these dense dynamic data clouds. And we're going to do an analysis of how that is deployed within the healthcare system, and then an economic analysis of how that affects the prevalence of Alzheimer's, of cancer, and all these other kinds of diseases. Another just interesting note on that front is that Washington State University just launched a new medical school. It's going to start this fall. They're putting every single one of their first-year medical students through Aerovale, through the scientific wellness program, and then it's going to get integrated into the curricula of what they're doing. And Lee and I and Ilya Shmulevich and Sui Wong at ISB are just about finished on a textbook on systems biology and systems medicine that's going to be utilized in that context as well. Okay, so that's just a little bit of context around how we can use how we can do longitudinal mapping to understand the early signs of cancer and Alzheimer's, and also how we can use genetic risk via dense dynamic data clouds to understand what might be warning signs in advance. Okay, I'm going to shift now away from that into some of the other work that we're doing. So we can look certainly for molecular diagnostics, early disease perturbations. Here's one that's quite interesting. This was done by Jocelyn Pearl, who's here in the auditorium as well. Jocelyn's a grad student who is graduating this year. She's fantastic. So if you need someone, you should try to compete for her. She's great. But anyway, she's done a lot of work here along with Seth Ahmet, who was a postdoc in the group, who's now a professor out at the University of Maryland. But in this case, this was an interesting study in mice. This is in Huntington's disease. And these were transcriptomes. So we did about 800 different transcriptomes during the very earliest stages in the mice. So this is 4 weeks to 20 weeks. So this is way before there's any symptoms, just to see if there was any kind of change that came. And one of the interesting things, and the idea here is trying to look at that earliest disease transition, just like what we're doing with the scientific wellness cohorts. And what you can see here is there's a few things that came out that were quite interesting. So here, this is a gene called PANK. And what you can see is that it's essentially not differentiated early on, but around, I don't know, the 10th week or the 12th week or so, we start to see a pretty strong divergence between the mice that have the extended CAG repeats that leads to Huntington's, which was discussed in the last talk, as well as compared to controls. And you can see that hold true for both black 6 mice and, to a lesser extent, in CD1. But the idea here is just basically being able to use the dynamics, in this case in mice, and in humans we can do these dynamics via the blood to see those earliest changes. I think I'll skip this really quick in the interest of time. This is only just to show for many different pathways. Basically, you just see that it's going from white to darker blue, basically, as you go forward in time. That's just showing that even early on, way before symptoms, we're starting to see more and more networks being hit by differences between having that extended CAG repeat in the HTT gene and not. And that's essentially driving all of that. If we shift back to cancer, this is a paper that just came out, I think, this month or last month in Cell Systems that we did joint with Lee Hood's group and Charles Cobb's at the Swedish Cancer Institute here in town. But basically here, what we did was to look for markers of glioblastoma, and so, which is brain cancer. And so here, we followed a trajectory, and I won't go through all of this, but basically, we did discovery proteomics, identified a set of proteins that were matched to the surface of the cell. So this is a cell surface specific or cell membrane specific signature. Identified 33 proteins that seem to be enriched along the in cell surface proteins for GBM. And then it turned out that when we looked into it further, we were actually able to find 4 of those that secreted into the blood very nicely and gave us very high accuracy in terms of being able to differentiate blood that came from glioblastoma patients from normal controls. I don't want, I'm careful to not say that's, it's not a biomarker yet or anything like that. There's a lot of specificity that needs to go through before we'd get there. But what's quite interesting that, but through this sort of very principled mass spec approach of identifying what's on the surface, what's very differential on the surface from GBM and not, we got very good validation there. The other element that we did is we used these 4 proteins in a more specific scenario in 10 patients, so small n, but this was just kind of the tail end of the paper after review. But basically, we got 10 additional patients where we were looking at recurrence. And so once the people were treated with GBM, as they watched as their tumor recurred, these proteins were also useful for, appear to be useful for monitoring that recurrence. Potential connections with neurodegeneration, because that's obviously the point of today's talk. And this is interesting. So these don't represent any super deep analysis on my part. This is me literature searching, but just to show a lot of connections. HMOX, which is one of these, turns out that is related to A-beta mediated hypomethylation, and it correlates also with cognitive impairment in Alzheimer's disease. Okay, so that's interesting. CD44 turned out, very simple literature search. It's expressed in non-myelinating Schwann cells, and it could play a role in neurodegeneration-induced glioplasticity. There was another one. VCAM1 there at the bottom. So this was just a quote taken directly from the paper. Endothelial activation, especially VCAM1, was of clinical significance in Alzheimer's disease. And that reflected macro and microstructural changes, poor short-term memory, et cetera. So I bring those up just in the sense that there's so many networks in common. And I think we're all also familiar with the fact that every gene seems to be linked to every disease in some form or another. So I wouldn't read too terribly much into this, other than to say it's clearly not just distinct players. There are a set of networks, and it was discussed earlier today in terms of trade-offs. And it's really true. There are many different trade-offs that every cell, every system in the body needs to make. And falling on one side or the other, you get into different disease scenarios. So it's not surprising that we see the same kinds of systems implicated over and over again in multiple different diseases. Anyway, so just a little bit about diagnostics. Okay, so now I'm going to shift gears again. I know this is a little bit like ping-ponging between Alzheimer's, neurodegeneration, and cancer, which is kind of what we've done in the lab. Now I'm going to ping-pong a little bit even further. I'm going to talk to you a little bit about honeybees. So basically, this was a study we did a little while ago, and it turns out that this is in some ways maybe the most centrally relevant to the intersection of cancer and neurodegeneration that we've done. So this was work that we did we published this in 2011. The reason that I'm going to say a little bit about honeybees is that they are really a key model system for the genomics of social behavior. And I got into this when back in the days when I used to be a professor at the University of Illinois in Urbana, and I worked with a fellow there, Gene Robinson, who was the director of the Institute for Genomic Biology, fantastic scientist, great guy. But basically, he had done a lot of work on different behaviors in honeybee and their relation back to transcriptomics. So he calls these neurogenomic states. And it turns out that there's a pretty strong neurogenomic state that's related to a lot of these behaviors. And he came to me at the end of this NSF grant that they had, and he said, you know, see if I want to do some systems analysis of this data. And he said they've done 20 experiments. And I was like, oh, well, that's kind of a start for systems biology. He's like, that's 2,000 transcriptomes across, 20 different massive studies, it turned out. And so I was like, okay, that sounds good. So without going into the details, we built a transcriptional regulatory network out of those data. And basically, what we were able to do is to build a model, a predictive model that could predict expression of genes in new scenarios, which I won't go into here. We mapped that via integrating statistical and mechanistic networks. So basically, we mapped that onto a metabolic network. And one of the things that came out to be quite interesting there is that we found that there was a metabolic shift in highly aggressive bees, so bees that are really likely to sting you. And it turns out that they run, essentially, aerobic glycolysis. And this was really interesting because this had not been observed, I don't think, before. We published this in Genes, Brains, and Behavior in 2015. But basically, this was a metabolic shift underlying a persistent mental state, essentially, of aggression. And the interesting thing about that, of course, is that aerobic glycolysis is, in essence, in cancer, what's known as the Warburg effect. Right? And you're seeing this great slide from van der Heyden again. But basically, this is very relevant to that. So what it said is that there are all these normal behaviors that are underpinned by similar metabolic differences as what we see often in cancer. And shifting then, this is from this nice imaging study here that shows where aerobic glycolysis appears in the normal human brain. And what's really interesting is that there's two connections to Alzheimer's disease here as well. So there's a decrease in total glucose consumption in the brain, including pre-onset. And this distribution of aerobic glycolysis in normal adults, it correlates spatially with where A beta deposition occurs in Alzheimer's disease patients. And so what was really fascinating about that then is that there seems to be then this connection between this shift into aerobic glycolysis and this amyloid deposition. It also relates very much to what we've been doing in the context of the Alzheimer's Pillar project that I talked to you about before, where the physicians that are working there really try to get the people to get into a ketogenic state, basically to try to shift away from some of these kind of metabolisms to try to have an effect on A beta and other pathways. Okay, I heard the bell, so I know I don't have too much longer to go, so I will apologize to the people whose work I'm about to zip through very fast. But basically what we're doing, so I mentioned we built transcriptional regulatory networks for honeybees. We now have a much better, more advanced way to do that, of course, because the world moves forward. But we have been building out an approach we call transcriptional regulatory network analysis, or Trina. So this basically leverages all the wonderful work that's been done by ENCODE and leverages all the big data resources that NIH has enabled, but essentially lets us build out transcriptional regulatory networks that use DNA hypersensitivity to find open chromatin regions, to identify footprints, to look for motifs within those footprints and so forth. But essentially, without going into all the details in the interest of time, what this is letting us do now is to build out for all the different genes that we've got, potential activators and repressors for all of them, and those are, of course, putative. You go through a huge scale down here in terms of the number of motifs you find in the genome, which is 2.3 million, and then filtering it down through many, many steps where you need multiple levels of evidence till you get down to a small number of targets for any particular disease. But basically what I wanted to say is just that We've built a big pipeline as part of our Big Data to Knowledge Center and working with great collaborators, Ravi Maduri and others at the University of Chicago. We now are near finalizing a pipeline that allows these to be done at high throughput, which has required actually a lot of computational effort. And then we will release to the public later this year a mapping of all these different networks for all these different 26 different brain regions, as well as other places in the body. So there's a lot of things that we can dive through there. So and that will be, I think, a nice resource for the study of both brain cancers as well as neurodegenerative disease. We did one of these type models for Huntington's disease, and this will be my last point. We identify a number of transcription factors that are affected them and their downstream modules. This work that was done by Jocelyn Pearl. Trina, I should say, has been done mostly by Corey Funk, who's here, as well as others. The downstream regulons have also been affected. So I should say, for one of these, we moved forward for validation for a number of reasons, it being really a top hit as well as having really good antibody. When we look at that, we're able to identify all these modules that are differentially expressed by the intersection with this SMAD3 gene that comes out as a key regulator in Huntington's disease. And again, just to be in the interest of this talk, when we validate this SMAD3 via ChIP-seq and we find that the model predicts accurately its effects in Huntington's, and again, we do the simple literature search and you find SMAD3 also implicated in TGF-beta signaling during carcinogenesis. So basically you can always find these kind of things, but there are a lot of interesting avenues, I think, to go down. All right, so just to come back then, full circle, we talked a lot, especially at the beginning, about this notion of dense dynamic personal data clouds, being able to dive in, understand that data using network analyses of different types. This is really the basis, we would say, for personalized medicine, for cancer and neurodegeneration. So to conclude, number of points. So cancer and neurodegenerative diseases, both been extensively studied. There's still a long way to go on each of them individually. The interface, I think, not very much. Not very many people. We've heard a couple talks today that have really done that, but we haven't done that a lot in the past. I think that as we start moving past 10,000 dense dynamic data clouds, we'll be able to identify early transition points for a number of people that will transition in cancers, which is already happening, as well as neurodegenerative disease, which we expect will also happen. We can use, as I mentioned before, just this genetic risk of disease along with the dense dynamic data clouds to understand how that disease risk manifests. That should be a pathway to early interventions to understand early events in neurodegenerative disease and cancer. We've already seen that very beautifully, I think, in Huntington's in the mouse models and so forth, but this amount of dense data in the blood should give us more insights into that kind of thing for human diseases. We can get more insight, I would say, into driving networks via being able to look at these networks that are being built and are getting better all the time because of the great work across the community. And that, of course, can give us better drug targets. So with that, let me just, I think I thanked most everyone along the way, but if not, just thank various people involved in this work. Corey Funk, who did a lot of the work for Trina. Seth Ahmed, who did a lot of work for Trina and HD. Andrew Magis, who's now at Airvale, who is who did a lot of the work for the Pioneer 100 study, along with John Earls. Jocelyn Pearl, who did all the HD work that I showed today. Paul Shannon, who did a lot of work on Trina, as did Roy Donovan, who's actually now here at the Allen Institute. And with that, I'm happy to take any questions. Thanks.
We have time for a few questions.
I'll shout. So, how easy do you think it would be to change people's behaviour? Because we all know that we know we shouldn't eat too much this, and we shouldn't smoke, and we shouldn't. And then people, and sometimes people, you give them a bad genetic result and say you're at risk of this, and say, what the hell? I might as well carry on doing it because I'm already at risk of it. Or somebody who's not told they're not at risk of it goes back and does the bad behaviour or the adverse behaviour. Or how do you think it's going to work out?
Yeah, I think that's it's a great point, and I usually talk about that quite a lot. Today I was trying to jump through a few things. So when I talk about the scientific wellness, when we did the Pioneer 100, one thing I didn't mention today, which I usually would, is we had a wellness coach that was associated with everyone. So that you take that actionable information, there's a personal relationship between them and a coach to work on behavior. When I mentioned that 170 people we have working over at Aravale, for example, probably 50 of them are behavioral coaches. So that's a very big element to what's going on. We could talk a lot more about that. In terms of engagement, I think that having the data is useful but not sufficient, for sure. A personal relationship with someone is definitely helpful. Trying to understand also the social environments and kind of what are the cues, I think that's also important, because changing human behavior is, as you know, very challenging. So we have huge efforts actually around that. And then the other element, though, that I also hope in parallel is that as we start to have a molecular understanding of these things in more and more detail, hopefully you will be able to develop preventives in more passive kind of ways that help to mimic some of those really good behavioral decisions and so forth. That's been, of course, very challenging to do because of the complexity. So I think there's a lot we can do on behavior, and then on a parallel track, I'm hoping that we can do more in terms of making molecular interventions better and better mimic the huge impact you can get from behavior.
Better and safer, because if you're going to persuade somebody to do something for decades to improve their health, so something that's incredibly non-toxic.
Incredibly non-toxic, that's right. Yeah. Well, and you would have integrated monitoring all the time too, so you can really monitor for. We actually think we're going to learn a ton about drug toxicities that aren't known today. That's another thing we think we'll learn a ton about. Yeah.
That was a very lovely presentation, and particularly interesting information about the bees there. So you started your presentation by saying how expensive it is, just the healthcare costs as they stand currently. What do you put the annual cost for each? So for an individual, were they to want to pay for the sorts of extensive studies that you're doing on them, what's that individual cost?
Right, so if someone comes into Aravale, for example, it's $3,500 for the first year, and then it's between $1,000 and $1,500 for subsequent years. There is one really low cost for $600 if they don't want to do all the testing. That is, I have to admit, a little bit below cost at the moment. I mean, that's somewhat subsidized by VCs. But not by a lot, and that number's coming down. The first year's, a lot of the expense of the first year is whole genome sequencing. And so, there are ways that could come down, right? You could do a SNP chip or something like that instead. But that's roughly what it costs today to do all the kind of things that I touched on a little bit. Those costs are declining, which I think is important. And then that is a big part of what we're going to do with the Providence study is really look at economic impact. One of the things I've argued at Providence is that if you can do that anywhere close to break even, in terms of basically what you've done is you've just gotten a cost reduction on jumping your healthcare system to the lead in terms of having a huge amount of data to drive precision medicine of the future, and you do it at a discount because of all the health savings that you get by harnessing value out of that data to help improve people's health and avoid disease. And I didn't get into, today I didn't really talk about disease avoidance, but in the practical, but there's a number of cases where that works really nicely. And so that economic analysis is going to be really key.
And obviously you haven't been doing this for a very long time, but once you have more data behind you, you could actually start to identify which tests would be key at which time frames, at what intervals. Because right now, when you hit 50, you get the standard colon screen and this sort of thing. Presumably out of this, you'll start to get data which indicates that you won't have to do the full spectrum on an annual basis, but there would be certain key things that you want to pick up on annually, and then maybe every 5 years you'd want to do this test.
That's exactly right. In fact, one of the things that we've been doing, or I should say that Aravale's been doing to bring down costs on that front is exactly what you're talking about. Like you find out, because as we measure all these things from people, you find out there are certain things that you don't really need to measure over and over again. They don't change all that often. There are others you find that you really want to have more fine tuning. And we're also, Rory Donovan was working on this when he was still in my group before he moved here. But one of the things we're doing is building out predictive models where you predict every single analyte from other analytes. Kind of a big Bayesian network kind of thing, although it's a little bit different than that. But basically, what you can do in that scenario is you can start to have a hierarchical network of, you measure these things, and then if something is wrong here, then it triggers it down. So you can bring down costs that way by having more tailored predictions. The other interesting thing is that it's really good for determining when you have a real signal or not. So if say if you've got, metabolite A and it's got the things that are connected to it in that predictive network, if you have a measurement, someone comes in and it's like it jumps way high, you can look at all the things that are related to it or correlated to it, associated with it, right? Because if none of them change, that's probably just a bogus measurement, right, which happens every once in a while. But if it seems to pull on the network it's connected to, then that makes you think, wow, something has really transitioned. It happened. So there's all kinds of ways we can get into that in interesting ways.
Okay, thank you very much. We're going to